Technetium is a very versatile chemical. It can be incorporated into a range of substances that are biologically active. The substances it is incorporated within then binds or joins or accumulates in the organs that are being targeted for scanning. So if we want to scan the bloodstream, then we incorporate technetium into a substance that will easily move around in the bloodstream. If we want to incorporate it into a molecule that's absorbed into bone, then it could be used as a bone scanning chemical. Targeting of organs with certain chemicals, of which technetium is one, and incorporated within, helps us to scan and diagnose disease. Radioisotopes and the substances that they're incorporated into are called radiopharmaceuticals, and they're chosen for particular reasons. And here we have a group of reasons, but they have to be easily circulated around the body in the bloodstream, so they need to move around to come to the organs that are being targeted. They need to be able to be absorbed or uptaken by the organ that we're interested in, such as the brain, liver, so on, thyroid. They have to have a short half-life, but it has to be long enough to see how the metabolism of this organ or this targeted structure works. But they also have to have a short enough half-life to minimize the radiation dosage that the patient receives. Here we have some radioisotopes produced in a nuclear reactor. Chromium to look at red blood cells, also in gastrointestinal protein loss, so issues with the intestines. Copper, often used to study genetic disease, such as copper metabolism. Iodine, which is used particularly to look at the endocrine system, or tumours, or even the kidney system. The human thyroid particularly absorbs iodine, so if you can put a radioactive traced iodine in there, it will absorb into the human thyroid, which is a gland in the neck, and it can be studied using gamma scans. Iridium, molybdenum, this is our source for our technetium-99m. Phosphorus, used in the treatment of excess red blood cells. Phosphorus is commonly used in bone disorders, but studying the bone and making bone scans, but which is where red blood cells are produced within the bones. Samarium, used with other substances, and we're looking at stopping metastasis of bones and reducing pain. Metastases are the spreading or finger-like projections of a tumour into other tissues. Technetium-99 is the most commonly used one, used to image the brain, thyroid, lungs, liver, spleen, kidney, gallbladder, blood, skeleton, bone scans, bone marrow, other glands in the body, the heart blood pool, and other infections. We have some cyclotron produced radioisotopes, so they're being produced by bombarding with ions, usually changing the number of protons, which is then results later in different number of neutrons and a production of a positron. So cyclotrons to do with positrons, positive electrons. And we have the common ones here, used in PET scanning. Fluorine, often used in FDG. Fluorodeoxyglucose, as tumours and the brain uses more glucose. Oxygen, 15, often used as part of the H2O molecule. More active parts of the brain often absorb more oxygen or have greater blood flow. So this water will go to those areas that require more blood flow. But you also have carbon and nitrogen. These are radiopharmaceuticals used in PET scanning, which again, PET scanning gives us an idea of the function of various organs by the way, or the uptake of materials into those organs. Whereas X-rays and gamma MRI scanning often produces just a structural image of what's there. The PET scanning gives us a functional image, how the organ is using the substances that have been radioactively labelled. Here we have a PET scan of a person, and you can see the hot zones. We have some areas here. We also have a large area here in the brain, but that could be just normal brain usage. But there could be a further examination of that. This is a PET scan. So we're looking at positron emission reacting with an electron to produce two gamma rays, sorry, not particles. That can be traced back to the position. So we have a number of issues in this patient. Some of these could be such as this one, the site of injection, where some of the substance is fresh in the injection site, trapped there. And this one uses a 18 fluoro, so it's a glucose molecule with fluorine 18 attached, which is a positron emitter. Other problems and issues in this patient, and we call these hot spots, where a lot of the radioactive tag has accumulated and you're getting more of the positron produced. As a result, you're getting more of these identifiable gamma rays produced. Also, from a PET scan, the resolution's not great, and you have attenuation, which means these gamma rays, sorry, not particles, they're photons of gamma, attenuate. They're not always equal in strength, and we'll see for PET scanning. But if this distance is greater than this distance, there's some loss of intensity of these photons compared to these ones. That would indicate that there's greater distance, or there is more tissue or something. The photons are being scattered as they travel greater distance than here. You can see if this person was in a PET scanner placed around them here, the distance between the two scanned gamma rays for this particular area, this one's greater than this one. So the computer can work that out when it receives these two gamma rays, gamma photons. It can trace back and see, based on the strength or the intensity that is read, can work out where in the body that tumour is actually occurring.